We're looking here at the prokaryotic cell, which is typical for bacteria. The very first difference between prokaryotic cells and eukaryotic cells is the size. The eukaryotic cell is way bigger than that of prokaryotes. The other difference comes from the fact that bacteria contains circular DNA. There are two types of DNA in the bacteria. There is the nucleoid, which is the large circular chromosome, and the plasmid, which may not always be found. Another difference comes from the fact that bacteria contain the 70S small ribosomes in comparison to the 80S large ribosome you find in eukaryotes. The third difference is the fact that bacteria contain some unique structures, such as those pili. Now, pili are largely involved in what we call bacterial conjugation. Bacterial conjugation is when plasmid DNA is transferred from one bacterium to another. And this is quite harmful for humans because if the bacterium on the left contains antibiotic resistance, then the bacterium on the right will gain this feature. Another feature of prokaryotic cells is the presence of flagella. Now, flagella are also found in eukaryotic cells such as the sperms, and they're meant for swimming. Bacteria may have more than one flagella. One last feature of prokaryotic cells is the presence of a cell wall. Now, the cell wall is made of a complex of proteins and carbohydrates known as the peptidoglycan. And we do have a slimy capsule, which is entirely made up of carbohydrates. The slimy capsule provides protection for bacteria against viruses and against phagocytosis with our white blood cells. In addition to that, the slimy capsule helps bacteria to stick to surfaces such as our teeth, and it also helps preventing dehydration in bacteria. One unique feature of all eukaryotic cells is the presence of the nucleus. The nucleus comes with a double membrane. In addition, it contains nuclear pores. There's one section inside the nucleus which is responsible for the making of large ribosomes. This is known as the nucleolus. The nucleus also contains chromatin. Chromatin is a complex made up of DNA and proteins known as histones. The DNA is capable of coiling and uncoiling around these histones, which would shorten the DNA and would form what we know as the chromosomes. The 80S ribosomes, or the large ribosomes, can be found freely in the cytoplasm, or they can be found associated with membranes known as the rough ER, or the rough endoplasmic reticulum. The rough endoplasmic reticulum is responsible for protein folding, and it's made up of series of membranes known as the cisternae. The smooth endoplasmic reticulum is responsible for the synthesis of lipids such as cholesterol or phospholipid, and is also made up of the same type of membranes found in the rough ER known as the cisternae. Golgi apparatus is largely involved in protein modification. Proteins are made on the rough ER and then exported via vesicles. They will fuse with Golgi from its convex side, and Golgi would be responsible for the modification. This protein modification involves the processing, packaging, and addition of carbohydrates to these proteins. The last organelle in this series is the mitochondria. The mitochondria is responsible for aerobic respiration. It has double membranes, and that inner membrane is folded into what we call cristae. These cristae are increasing its surface area, so more ATP is made by aerobic respiration there.